for this last day, seventh day meeting, a refresher course. So today also we have an exciting panel discussion and three lectures, three talks, and the panel discussion I'm sure is going to be an exciting and very beneficial to all of us because we have uh, a very experienced, personnel scientist in the area of plant biotechnology and allied sciences who have inspired, of course, many of us, young minds and many of us, and they hold a very high position currently or before in India or abroad. I feel privileged to introduce very briefly the respected panelists. Divya, can I have their profile? Yeah, I'll take only a couple of minutes for each of you. Dr. K. Narayanan, the moderator of this panel discussion, is currently is managing director of MetaHelix Life Sciences, a Tata enterprise. MetaHelix is based in Bangalore and is focused on crop improvement using all the modern tools of plant genetic manipulation. Dr. Narayanan is trained as a plant breeder and molecular biologist. He graduated with a PhD in plant breeding and genetics from Tamil Nadu Agricultural University, Coimbatore. He later on carried out postdoctoral research at Stanford University under a Rockefeller Foundation fellowship. Returning to India, he started his career as a scientist at SPIC Science Foundation, Chennai, and later he moved on to lead a crop transmission and functional genomics program at the Monsanto Research Center located within Indian Institute of Science, Bangalore. In 2001, he, along with four other scientist colleagues, founded MetaHelix Life Sciences and has since been leading its operations. Dr. Narayan is a member of several prestigious committees and advisory bodies, including the several scientific task force on agri-biotechnology of the Department of Biotechnology, Government of India, and the Vision Group on biotechnology set up by Karnataka government. Uh, can we have the next panelist profile? Yeah, Professor Sudhir Sapori is currently Emeritus Senior Scientist at the International Center for Genetic Engineering and Biotechnology, New Delhi, where he also worked as ACRB DSC, Government of India Distinguished Fellow, Group Leader, and Director. He obtained his PhD in 1973 and postdoctoral or visiting scientist stints in Germany and USA. He earlier worked as a professor at the School of Life Sciences, Jawaharlal Nehru University, New Delhi, where he also served as Vice Chancellor for 5 years. His major area of research has been to understand the stress and light-mediated gene expression in plants. His group discovered the role of galaxylate enzymes of a neglected biochemical pathway in plant stress physiology. He has edited 13 books and published over 260 scientific papers in internationally reputed journals and about 60 book chapters. He is a fellow of Indian Science. He's a fellow of all the prestigious national academies, Indian National Science Academy, Indian Academy of Sciences, Bangalore, Indian National Science Academy, India, Allahabad, National Agricultural Academy of Sciences, New Delhi. He is also fellow of the most prestigious, the World Academy of Sciences, Trieste, Italy. He was vice president of Indian National Science Academy. Sorry, I've already covered that. Yeah, amongst others, he has been conferred National Civil Award, Padma Shri, and S.S. Bhatnagar Award of CSIR, uh, Birbal Sahni Medal of the Indian Botanical Society, and the Lifetime Achievement Award of the Indian National Science Congress, Indian Pharmaceutical Society, Biotechnology Research Society of India, and Indian Botanical Society. He was the first Indian to receive the earned Macrobi corresponding membership of American Society of Plant Biologists. He has been conferred Doctor of Science degree honorary cause from Banaras University, Varanasi, and Rani Durgavati University, Jabalpur. He was the president of Indian Society of Plant Physiology. Currently, he is the president of Biotech Research Society of India and Vice President, DAV College Managing Committee. Next one, please. Next profile of next panelist. Professor Ajula D could not join, yeah, because of some health issue. So uh, let me briefly introduce Professor K.C. Bansal. Professor K.C. Bansal, currently Secretary of National Agriculture Academy of Sciences, and he was former Director, Indian Council for Agricultural Research, National Bureau of Plant Genetic Resources, New Delhi. And Dr. Bansal, he obtained his doctorate with gold medal from Indian Agricultural Research Institute, New Delhi, and pursued postdoctoral from Harvard University, I think in Professor Bogarat's lab, if I remember correctly, in Cambridge, USA. He worked as visiting scientist at Rutgers University, USA, under the aegis of Rockefeller Career Biotech Fellowship. Currently, Professor Bansal is adjunct professor at the Center for Crop and Food Innovation, Murdoch University, Perth, Australia, and served on the board of directors of Global Plant Council, board of trustees of MS Swaminathan Research Foundation, Chennai, and on the Research Advisory Council of IRI, USA, New Delhi. Professor Bansal served as elected secretary, I've already mentioned, elected secretary of NAS, as director of Premium Eyesore Institute, that is NBPGR, and elected vice chair from Asia of the 15th regular session of the Commission on Genetic Resources for Food and Agriculture of the United Nations. Professor Bansal played a key role and visionary role in formulating World Bank project on national agriculture higher education, which is implemented across 75 agriculture universities in the country since 2017. Can I have next one? Yeah. Dr. Vibha Hauja, currently she is Chief General Manager of Biotech Consortium of India Limited, New Delhi. And she began her career in 1993 and is an expert on biosafety and regulatory aspects, particularly with reference to genetically modified organisms, having more than 30 years of experience in this field. She is very well versed in issues related to Indian biosafety regulatory frameworks and has been part of the formulation and dissemination of guidelines, also served as a member of expert committees. She has expertise in planning and providing support in regulatory compliance for taking forward technologies for commercialization. She is also actively engaged as a resource expert on gene-edited plants in this region. In the region, throughout her long and distinguished career, she has been actively involved in biosafety capacity building initiatives in India, throughout South Asia, South Asian countries like Bangladesh, Bhutan, Sri Lanka, a few other countries. She has led several capacity building activities in the area of modern biotechnology, imparting training through seminars, workshops, national and international training programs, and preparing well research publications of various stakeholders related to biosafety and biodiversity issues, etc. Yeah. Dr. Raman Sundaram, currently director of Indian Council for Agricultural Research Institute, Indian Institute of Rice Research, Hyderabad. He obtained BSc in Agriculture from Tamil Nadu Agricultural University, Madurai, MSc in Agriculture in Molecular Biology and Biotechnology from GB Pant University of Agriculture and Technology, Pantnagar, and PhD in Plant Science at University of Hyderabad. He was IUSSTF postdoctoral fellow at the Iowa State University and Cornell University, USA, and USAID or ABSP research fellow at Cornell University, USA. He served in various positions as scientist, senior scientist, principal scientist in India, ICAR Institute, Indian Research Rice Research, and currently working as director of this premium institute. Dr. Raman's major research areas are crop biotechnology, rice genetics, and molecular breeding. He received several awards and honors like DBT Award for Biotech Product and Process Commercialization, PN Bell Award for Millennium for Excellence in Crop Science, ICR IR New Delhi, CSR Award for Science and Technology Innovation in Rural Development, Hari Home Ashram Trust Award, ICR for the Biennium 2010 and 12, among several others. He is a team member in our expert crop group for particularly rice for developing DNA fingerprinting protocol constituted by ICR, external subject expert for plant biotechnology at Kerala Agriculture Institute, member expert committee on crop molecular breeding, DBT, among others. He was chief editor of Journal of Rice Research, editor of Journal of Plant Biochemistry, Biotechnology, and some other journals. He is a fellow of National Agricultural Academy Sciences, New Delhi, Indian Society for Genetics and Plant Breeding, and Indo-US Science and Technology Forum, USA. So with this, I think, yeah. So now I request Dr. Narayan, the moderator, to conduct the proceedings. Dr. Narayan, please.
Thank you, Professor Rajam. Can you all hear me?
Yeah, very well.
Okay, and also thank you for the detailed introduction of me as well as the different panelists. But I must point out that I am an academician turned entrepreneur. First 15 years of my life was in academics, teaching PhD students and all that. Yes, MetaHelix was one of the companies I founded and ran it for about 15 years before we divested it to the Tata. So now it's a Tata enterprise altogether. So I was the managing director of MetaHelix Life Sciences. And since then, I founded a couple of new companies, one of which was again divested to Tata Consumer Products Limited. I continue my engagement with academia and the industry through positions in several of the boards and also in several of the committees. So that's in short. Good. So we have a very high-powered committee, our panelists here. We have Professor Sapori, the Vice Chancellor of the prestigious, was the former Vice Chancellor of the prestigious Jawaharlal Nehru University. We should have had another Vice Chancellor also, ex-Vice Chancellor on the panel, Professor Arjuna Reddy, but I learned that he is not keeping well. So we wish him speedy recovery. We have directors, one former and one current directors of two national institutes. Professor K.C. Bansal was the director of NVPGR, and Dr. Sundaram, a good friend and a very eminent dynamic director of the Indian Institute of Rice Research. And we have Dr. Vibha Ahuja, who represents a quasi-government organization, the Biotech Consortium India Limited, a bridge between the public and the private industry, if I may say that. I'm trying to put it in little few words. She does many things, in terms of creating awareness of regulatory systems, runs a newsletter. So I dare say that between the panelists who are present in this meeting, we have an experience of over 200 years in science administration, and science dissemination. So with that, I would just like to lay out my thoughts on the format for this panel discussion. We would like to start with each panelist making their opening remarks on the topic biotechnological interventions for sustainable agriculture. And then we can have a freewheeling discussion, not just among the panelists, but also with the audience. I learned that a lot of people have joined online. So if you have, in the interest of hygiene and keeping things under tight control, if you have questions, please post it in the chat box. I will once in a while check that out. I will also request the organizer, specifically Professor Rajam, to, during the interactions with the audience, if they can take out key questions from the chat box. I will also try to look at that and keep it ready. And as a matter of hygiene, I would request all the other participants who are not speaking to keep their microphones in the mute mode unless you are called upon to make an intervention. So let's start the opening remarks. We are already about 18 minutes into this panel discussion. I will start with the former Vice Chancellor of the Jawaharlal Nehru University, Professor Sudhir Sapori. You heard a lot about his accomplishments in the introduction, but I must add that he was one of the initial pioneering researchers from India to work on the molecular basis of stress tolerance and trying to introduce stress tolerance. I still remember his work with the proline biosynthetic pathway, the trying to introduce the P5C gene when he was in ICGB and trying to get salt resistance. And these things are becoming very, very important these days because of climate change and the increased incidence of these stresses that is confronting our agriculture production and agriculture quality. So Professor Sudhir Sapori, I would request each of you to keep your initial remarks to about 5 to 10 minutes, not more than that, please. Thank you. Professor Sudhir Sapori.
Thank you, Dr. Narayanan, and such a pleasure to see you after a long time.
Thank
And
you.
I'm really privileged to be amongst the esteemed experts over here. First of all, thanks to Dr. Rajam, I think, for organizing this wonderful workshop. And it was pleasure initially on the inauguration day to listen to Professor Pakiretti, Dr. Redama, and Venkateswarlu and others. Unfortunately, because I am in US, I could not listen to many lectures, but the morning session, I could attend many of the lectures, although this was getting late into the night. But it was very, very nice to listen to some of the speakers in this topic. Now, as was mentioned, the topic is basically on biotechnology and sustainability. And I think these are the two keywords I'm taking from this thing where we need to discuss about it. Of course, everybody knows the broader question in our mind, and that is whether we are able to feed the ever growing population. And that was one thing I found in all the lectures which were here. They would start with that. Can we do this? Can we feed the billion population on this planet over a period of time? And that too under the climate condition changes which are taking place all around. Now, I would concentrate a little bit on the word sustainability here. Now, to me, sustainability means maintaining balance. Now, balance between three or four points which I'm mentioning. So, maintaining balance between production and productivity versus population. This is one balance which we need to maintain. The second point is to maintain balance for sustainability purposes between chemical agriculture, which includes fertilizers, pesticides, and other agriculture practices vis-a-vis environmental safety. That is the greenhouse productions, gases, and all that. And the third sustainability things which we need to look at is the availability of agriculture land area versus urbanizations and forest cover protection. So these are basic ones, but there could be one could list many more. But essentially, what we are talking is to making a balance between gains versus losses, and that is what sustainability in the longer term one means. Now, people, there is a famous proverb which says that every gain comes with a loss. Now, the problem is that we cannot afford to have loss in our agriculture practices over a period of time. So, is it possible to bring about this sustainability under climate conditions? So, that's the question which you're asking. And President of American Society of Plant Physiology, in one of his articles, says that I'm very optimistic that this should be possible to do it. And his optimism flows from the fact that the greater advancements which are going on at the moment in the field of plant biology, crop biology, horticulture, and so on and so forth, gives you some kind of a confidence that probably you will be able to beat the impact of climate change and bring about sustainability in agriculture production systems. So, what is the way forward? Few points which come to my mind, and this is what everybody will be talking about, is to use genomic data with artificial intelligence very precisely for generating variability. I'm repeating, genomic data with artificial intelligence to generate variability. And we have now very, very good AI platforms. The very recent one, which was announced a week back, is Evo 2 data, this AI platform, which can now be used. So breeding, as we know, has always been in the process of selecting variations. Initially, we are selecting variations through natural variations, and now we are going advancements in the same thing through the studies of phenomics and also study of pangenomics in order to identify the variations in the natural populations, finding out the right kind of alleles, and so on and so forth. The other thing which we have been doing is to creating variation, and this has been done initially through random mutagenesis. And now there are very good techniques which people are still using. As there was a paper in sorghum, but they had generated 150,000 mutants. And by this new technique of PCR, they were able to identify variations in specific genes through a PCR methodology. So this is another technology which has been used to generate variations. And now the third thing which is coming up to generate variation is the genetic engineering and genome editing technologies. So basic thing which we are now moving with this lot of data which has emerged, the big database available, to move from, earlier we used to move from phenotype to genotype, now it is from genotype to phenotype. Whether you can identify the signatures of a specific phenotype through these genetic engineering technologies and functional genomics things, and then move into a specific phenotype. Now, once we have generated a lot of variability through different methodologies, either identified in natural populations or generated through different technologies, what we are moving fast is in terms of the breeding technologies for identification of specific traits in order to bring about homogeneous population, or what I call the development of the varieties of a specific interest, any variety of a specific trait or an interest. And for this, the way fast forward technologies are molecular breeding technologies. People are now going into haplotyping, rapid generation advancement technologies. So there are technologies for generating variability now, and there are technologies and good methodologies by which the selection of the specific trait from this variation can be fixed and a variety can be developed. So what we have to go in future is to bridge scientific discovery with practical solutions. And that is very important. This gap has to be the minimum gap if we have to beat and get into the sustainability things, bridging scientific discovery with practical solutions. Now, to move into the sustainability into this direction, I think what is very important is to develop transdisciplinary approaches. Now, these transdisciplinary approaches will be able to that will be able to find the blind spots and find solutions to those blind spots. And therefore, we also need to go into whole field biology studies. I think right now most of the genome editing and other things are going into the laboratory conditions or greenhouse, but this everything has to be done into field biology. And therefore, field biology, you will have to see the interactome. I'm calling it the interactome from this perspective, interactome of plants, interactome with soil. Soil, you are talking about water, nutrients, and microbes. These three things are most important, along with the environment. So plant, soil, and environment interactions have to be tested under the field conditions of any about the variability that you develop from either platform which one is going to use. And later on, I think we have to see whether these varieties which you have developed are able to uh, stand up to the climate changing situation. And therefore, the targeted approach will have to be to develop right climate models in order to see that what you are heading for. I think that's important. We don't have good regional based climate models, which needs to be there. We develop variation, but I don't know we variation from which soil, which environment and which climate which we are talking about. And in addition to these technologies, I think we, as everybody knows, go into the alternate crops, focus on the root phenotyping, nanobiostimulants and other things which can be talked about. So broadly, what I'm trying to say that we have to take a holistic view of agriculture practices, and this is both plants, animals, fisheries, and everything. And we need to bring about policy changes, economic factors, support funding from the government in order to reach our target, and that is sustainability in agriculture or feeding the larger population on the planet. Thank you very much. These are my brief initial remarks.
Thank you, Professor Sapuri. I think you timed it so well, 10 minutes. And you made some very, very important points. Genomic data and artificial intelligence for creating variation, the importance of molecular breeding, having a transdisciplinary approach. I cannot agree with you more. Agriculture today is the confluence of multiple technology domains. It's not just breeding or agronomy or botany. And the importance of field biology. Some of these points certainly are worth discussing or probing more. Let us circle back into it later during the panel discussion. Now let me move on to the lady in the panel, Dr. Vibha Ahuja. The platform is yours for your perspective on biotechnological interventions for sustainable agriculture. Please go ahead.
Thank you so much, sir, and thanks to the organizers for including me in this panel. Dr. Sapori has really set the tone, with the kind of, the need for all kind of approaches if we have to achieve the sustainability in crop production. So while, as you said, we can't agree more than this, we really need a big toolbox for the practical applications to, for achieving the crop improvement, increasing the productivity, and so on and so forth. So I would like to move on from what sir said, to the challenges which are there when you do all this work in the lab and you are not able to take it to the farmer's field. This is what has been happening with some of the biotechnology interventions, and where, we really need to all work together. This training course itself is like one of the steps, I would say, in to contribute to those issues. So, sir, I would like to move on and share some of the experience that we had in last 2.5 years. We've had a very positive approach. We are seeing after a long time, the new regulations which came in India. I want to keep myself restricted to the Indian situation for gene editing. We also saw a lot of field trials coming in, from the public sector institutions and so on. But still, and we organized actually more than 15, interactions at the grassroots level with agriculture universities, with the user groups like the feed industry, with the ethanol production industry, and with the, with the textile industry also at some point. So what we learned from all that is that, there is really a need for communication, communication about this new biotechnology interventions, moving on to the user groups is extremely important. People still are not, even within the universities, the potential of gene editing, the compliance to the new regulations, how easy they are, but they're very important. And all these things are really the areas which we need to address in addition to the research activities that are being funded or that are being, initiated across the country and also in many other countries. As sir pointed out, we've been working in other countries like Bangladesh, even Thailand. We played a very important role in making sure that they adopt good gene editing regulations, in addition to countries in the South Asia region. But what we see is a very, very big communication gap. While on one hand we talk of there's a little dichotomy in the sense that, like, for example, sir mentioned about the big data, the biologics, and all that being available, and we have to make use of that for sustainable agriculture. But at the same time, there is another big area of discussions which is on digital sequence information access and benefit sharing data. So the IP, biosafety, regulatory, all these issues have to be dealt with together for communication to the stakeholders, so that we can whatever the ideas scientists have or they develop in their laboratories can actually reach to the society for the benefits. So this is broadly I wish to communicate to start with, that don't leave these things out. They are very important if you want to reap the benefits for the farmers, for our farmers, and for the consumers. So these are extremely important issues to be dealt with, and not only for our scientists, but not only for other stakeholders, but our scientific community itself. Uh, I will just take one more minute to tell you, I'm really surprised at that we've got such good, regulations, which should be highly motivating to our institutions. But there's an important area of our institutional biosafety committees who are going to take the decisions. So they also need to be equipped and informed about these new regulations and how to deal with it. So let's not leave these areas out because the technology advancements are happening so fast. But the other system, the communications, the IP, the other things also have to cope up with that. So these are some of the areas which I wanted to highlight for the panelists to take note of. Thank you.
Thank you, Dr. Vibha Ahuja. I think you laid out a few challenges in leveraging biotechnological interventions or methods for sustainable agriculture in the country. And one among them, you alluded to the lack of communication. You put it as lack of communication. I guess it's a lack of effectiveness in the interaction between academia and the industry, if I may put it that way. You also mentioned about the regulatory system. And while we have a overall good system, there may be components on it which may need a reform. Like I talked to Sudhir Sapori also, let us circle back to some of these issues as we go forward in the discussion. So please collect your specific thoughts on the 2 points I made, industry, academia, interaction, and some aspects of regulatory systems which need some reform. Let me now move on to Professor K.C. Bansal, a fantastic researcher, research administrator. As I mentioned earlier, he created a lot of changes in the way academia interacts with industry when he was director of the NBPGR. Now, Dr. Bansal, for your opening thoughts on biotechnological interventions for sustainable agriculture.
Well, thank you very much, Narayanan. It's always a pleasure to be having discussion and, with all other eminent panelists, particularly Professor Sapori, with whom I keep interacting, quite frequently. And of course, also Dr. Vibha Ahuja, and nevertheless, Dr. Meenakshi Sundaram, our good friend, really doing wonderfully well. So it's a pleasure. And thanks to Dr. Rajam, and to the FABA, Dr. Adanna and others to give me this opportunity. I will actually take over from where Professor Sudhir Sapori, left. And I think he has conveyed a very deep message to not only all the young researchers, students, for all of us as well, how important it is to develop any kind of a comprehensive, holistic technology, for dealing with agriculture today as we are talking about it. And today we are talking about it, of course, climate resilience and sustainability, both as Professor Sapori rightly mentioned. And the technologies available are twofold, actually, when we talk about it, dealing with the current situation of agriculture. And that is, we have farming practices, through agronomic practices, regenerative agriculture, climate smart agriculture, as they talk about it, where they mention about increasing productivity, reducing greenhouse gas emissions, and not only reducing the emissions of the greenhouse, and therefore curtailing the temperature, but also even having some approaches for efficient utilization of some of those nitrogen or other fertilizers and natural resources which are contributing to greenhouse gas emissions, and capturing the carbon and bringing and sequestration back into the soil. So when we talk of all these technologies, very, very important, but one of the factors what I see is the most important factor of this technology is developing a new seed. As Professor Sapori also reminded, to developing these climate resilient seeds, which are multi-stress tolerant, having high yield potential, and also resistance to many of those pests and diseases. And friends, for the last over now, almost, let's say 100 years, we started with conventional breeding, we move on to molecular breeding, we move on to transgenic breeding, and we are now talking about breeding by genome editing. So these are some of the technologies, and all are important. I mean, sometimes you wonder that if the technology is there, it's like, working like a bullet and, kind give you the answer to all those, problems. It's not really that. I think, when we talk of a comprehensive way of moving forward, is also use of all these technologies in an integrated fashion. Conventional breeding, molecular breeding, transgenic breeding, or breeding by today, as we're talking about genome editing. So I will actually focus rest of my discussion on genome editing, particularly, and also not to undermine the importance of genetic engineering GM crops. While we have not been able to, unfortunately, it should be very clear, even to the young minds, we have not been able to use this technology to the fullest way as many other countries around the world have done it. There are 72 countries now dependent on GM crops and 206 million hectares under cultivation of these GM crops. But nevertheless, we are still struggling. This is not a very healthy situation. We must be very clear and understand it. Not we can say that everything is going good in our country. With regard to technology adoption, we were very wonderfully doing well with GM, BT cotton. But after that, we have not been able to crack and use this technology forward, while we continue struggling on almost on a daily basis, right from down to the communication to the judiciary levels, etc. But in the meanwhile, fortunately, as you all know, genome editing has come up in a big way. And genome editing, again, is people talking about three different categories: SDN1, SDN2, and SDN3. I'm sure this has already been covered in your lectures, but I would only like to make an emphasis upon here is that one should try to make use of fullest SDN1 and not as much as SDN2. Again, I say that SDN2 is important, but its efficiency is very, very low. The methods availability are not as many available as you would like to have it for different crops and for different traits. And the world over, 97% of genome editing is being done through only SDN1, which is the easiest way. And therefore, you can mutate or delete the function of a given gene, which is deleterious, which is causing disease susceptibility, which is having a negative impact on growth and development, seed development, etc., etc. And if you go around the literature, based on the genomics data, as Professor Supuri mentioned very clearly, we have a lot of genomic data available, all these artificial intelligence tools. If you go around it and look for those genes, even crop specific, you'll find plenty of them. Those genes still, we need to discover where we can apply SDN1 technology to improve a particular trait. I'm talking about it because this is given already results. Crops have been commercialized, in the world, at least, half a dozen of them, using this SDN1 technology. And we can have those genes, even orthologous genes from different species can be, tried in some of the crop species where such genes or genomic data, for example, are also not available. So there is a way out to go for. So I recommend, SDN1 there for this. I will not, again, to be very clear on this, not recommend clearly SDN2 because of various reasons. And we can talk about it later. And another factor which I would like to recommend here while talking of genome editing is there are more approaches of base editing, prime editing, or activation of gene expression, or repressing the gene expression, or epigenome editing. These are some of the new variations. But among all this, again, I would give major emphasis on prime editing. Base editing is again wonderful. There are results, particularly for wherever you need a point mutation targeted in the exonic region, or in the promoter region. That's another thing which we have to learn. What area, what part of the whole gene we like to, really speaking, go for editing. Because even you can do for particular resistance, there are certain effector proteins from some of those, bacterial species which go and bind to the promoter regions, as has been shown in case of rice, sweet gene promoters. So those effectors, transcription factor like effectors will be binding to those promoters of sweet genes, number of them. So if you mutate that promoter, and then those vectors will not be able to bind, will not be able to express those sweet genes, so that they will not be able to make more sucrose or transporters, which are important, for these bacterial species to thrive upon. So therefore, one should try to understand that as well, which part of the gene we would like to, really speaking, mutate. And where I'm saying prime editing, because sometimes, base editing you can only do to a certain extent, which is again important for herbicide tolerance, particularly, as has been demonstrated. But prime editing is something which the whole world is talking about today, that how can we use it more effectively? And it's got a better efficiency of transformation or editing efficiency than even SDN2. Which we're talking about. SDN3, by the way, I'm sorry I didn't mention much about it, because SDN3, as per our Indian regulation, it is as good in most of the world as well as GM crops, which is subjected to biosafety regulation. And I think quickly, before I go further, I've got one more message to give about identifying those target genes, etc. But before that, I would just like to mention, fortunately, in India, while we are not doing as much as before in case of GM crop, but we must thank profusely our policymakers, Government of India, to allow us exemption of SDN1 and SDN2 genome category, in case of plants. And therefore, we are really able to make much headway. We're able to develop a product which is undergoing field trials, both in rice. Dr. Sundaram will talk about it. One of his institutions has done wonderfully well. Dr. Vishnathan at IRI. These two crops, of two particular, events in rice, for two different traits are already undergoing All India Coordinated Research Improvement Program field trials, and which have been in a way approved already by our regulatory system, which is quite eased out. And therefore, I will request all those participants, those who are here in this particular webinar, to go through the standard operating procedures which have been developed, notified, which was notified on actually in the month of October 2022. And you must grab a copy of it like a Bible. Those of you who are really interested in business, kind of developing, genome edited products. So, but then the last point, which I want to make quickly, another 2 minutes, Narayanan, is this, or 1 minute is this, that we must try to integrate, our genome editing process with gene bank accessions, because that's what, as Professor Sapori mentioned, there's a natural genetic variability. We need to discover that. And people today are talking about it. Of course, functional genomics, even using CRISPR, therefore need to be given much emphasis upon, until unless, and many of the crops and traits we're talking about it, we don't have identified those target genes yet. So therefore, youngsters, you may like to therefore go into it, kind of excellent genetic variability, which is available over in gene banks, second largest in the world. We must try to explore it. You must try to see that we have got those new genes identified, alleles identified, which you can install in popular elite cultivars. Not in any cultivar of the crop. I may like to understand or emphasize this, in elite cultivars of any particular crop. So this is what we can talk about it more, discuss it further, but this is a technology I think we should be taking full advantage of it, as many other countries, particularly China and US, making inroads into this. Thank you very much, Narayanan, for this opportunity. These are my opening initial remarks.
Thank you, Kailash. I think you covered a lot of ground. You started with climate resilience, then went on to elaborate on genome editing. I know that you were a key person involved in putting together the guidelines for genome edited crops, and you have a lot of things to say. I already have some questions on the chat box on genome editing, so we'll circle back to that. Meantime, I think I will let the platform to Dr. Sundaram to give his opening remarks. Dr. Sundaram heads the Rice Research Institute, the largest food crop in the world, if I may say that. So Sundaram, your perspective on biotechnological interventions for sustainable agriculture.
Sir, thank you very much for the opportunity. When we talk about sustainable agriculture, what we actually mean is that we need to enhance the production and productivity in a sustainable manner with least damage or no damage to the environment. And the productivity should be sustained. Increase in productivity should be sustained. So keeping this in view, we also need to understand and then appreciate several challenges. For example, in the rice sector, presently we are comfortable in terms of rice area, wherein we have got more than 45 million hectares of area in which rice is cultivated. But the apprehension is that there'll be a significant reduction in area from 45 million hectares to about 35 or 38 million hectares in the next 30 years. So are the challenges like population growth, soil degradation, and most importantly, reduced availability of inputs like fertilizers, water scarcity, etc. And we also need to save the environment in terms of reducing greenhouse gas emission from agriculture, particularly from rice cultivation, while at the same time minimizing the biodiversity loss and minimizing the use of chemicals. And biotechnology is the only option for breeders in terms of bettering the agricultural practices, in terms of addressing whatever concerns I just enumerated. And the biotechnological tools, which I have visualized in terms of in rice sector, are precision breeding or molecular breeding, which has got mutated nowadays to genomic-assisted breeding, then genetic modification or transgenic breeding, and of course, along with gene editing. And finally, last but not the least, genomic studies, both structural and functional genomics, are very integral for application of biotechnological tools. When I joined ICR, Indian Institute of Rice Research, in those days it was known as Directorate of Rice Research, I joined after completing my master's as a DBT fellow at GB Pant University of Agriculture and Technology in molecular biology and biotechnology. I joined with a lot of enthusiasm, and at that point of time, I wanted to work on GM rice, wherein there are a lot of developments and wherein there are a lot of research papers. But fortunately, my mentor at that point of time, Dr. N.P. Sarma, Dr. Nukala Padmanabha Sarma, didn't allow me to work on genetic engineering or transgenic breeding. He made me work, or he forced me to work on marker-assisted breeding or molecular breeding. His was a master stroke in the sense that I could make significant gains or inroads, not only in terms of gene identification and better utilization of genes for biotic stress resistance, traits related to heterosis, and also other gainful traits like fertilizer use efficiency in the last 26, 27 years working here at IARR. And we have made significant gains or inroads with regard to molecular breeding. But in terms of what you call as the other biotechnologies like genetic engineering and functional genomics, it's my humble opinion that we need to do a lot more. Part of the problem are the government policies in terms of promoting certain biotechnologies and then limiting access to certain biotechnological applications. These are all indeed real issues we need to address. And another important thing which I would like to mention here is that in addition to leading IARR, wherein our main focus is on improvement of irrigated rice cultivation system, I'm also serving as coordinator of All India Coordinated Research Project on Rice, in which there are about 4,500 centers who carry out rice trials across the country. And we also have 100 voluntary centers. So one intervention which we have brought together collectively as team crop improvement in our system is that we are rather trying to mainstream biotechnology in the breeding process, wherein for the last 15 years, we are successfully conducting special trials for near isogenic lines or products developed through molecular breeding, wherein they need not undergo a rigorous testing along with all other entries. They need to be compared only against the recurrent parent or the variety wherein that particular trait or a few traits have been modified. And another major intervention which we have brought is implementation of genomic selection in the breeding program. For the last 2 years, we have been very diligently fingerprinting each and every entry nominated for ACRIP trials. Last year, we did it for 762 entries, and this year, the numbers are more than 900. So each and every entry is being fingerprinted with medium throughput rice SNP chip called as 1K Rika rice chip, which is actually helping us to understand the amount of diversity which enters into breeding program from different locations. And it is also helping us in terms of fine-tuning our genomic selection models and then pipelines. And this is something wherein I'll urge that it should be practiced with other crops also. And we are also slowly moving towards integrated genomic selection, wherein genomic selection is gainfully combined with speed breeding and also application of artificial intelligence tools for understanding and then interpreting both phenotype and then genotype data. And in this regard, one important point I would like to mention is that as a group of frontline policymakers in biotechnology, we also need to give adequate importance to functional genomics, because we started with a bang in terms of rice genomics initiative, wherein we were able to sequence, participate in the International Rice Genome Sequencing Project, and we were able to sequence a segment on chromosome 11. But my opinion is that we have not made any significant inroads or gains in terms of functional genomic studies, which are very, very vital for undertaking proper, identifying proper targets in terms of gene editing, but also in terms of rapid candidate gene identification and then deployment of the favorable alleles through molecular breeding. And another important area wherein we need to make sufficient investment in terms of both research and infrastructure in terms of pangenomics, wherein, for example, China in this particular field has sequenced more than 8,000 accessions of rice, and they are able to understand not only the range of allelic variation and the range of genetic variation which is available in nature, And this helps them to not only to fine-tune their gene editing strategies, but also rapidly access the variability which is already present and then deploy them in the breeding programs. And I believe that we need to invest sufficiently in the area of pangenomics of major crops, including rice. And one important or very happy note is that despite having spells of drought for the last, say, 5 or 6 years, we have indeed sort of climate-proofed agriculture in India, because I would like to just take the mention of rice. In the last, say, 10 years, the trajectory has always been in terms of significant increase. For example, by the year 2012, we were able to produce only 98 or 100 million tons of rice. And there have been significant spells of favorable and then unfavorable years in terms of climate with regard to rice production. But the rice production and productivity has not decreased. It has been steadily increasing despite the unfavorable years. And part of it is because it is estimated that in the country, more than 3 million hectares are under biotech crops. Of course, cotton is one typical example, but so are the cases in terms of maize-derived products in rice, chickpea, and other crops. And I believe that if not a major contribution, at least a significant contribution in terms of climate proofing agriculture in India is being taken care by our biotech crops. And another important debate which I would like to mention here is that among the policy makers, it has become a fashion or fad to compare transgenic crops versus gene-edited crops. While
1 minute, Sundaram.
Yeah, yes, sir.
1 minute, please.
Yeah, I'll just wind up, sir. One important thing what we are witnessing is that there's a debate that we should move away from GM crops or transgenic crops, and we should go full steam ahead for gene-edited crops. Gene editing is a very powerful technology, and we have demonstrated in terms of improving the yields, climate resilience of Samba Masuri. But in terms of as a tool for genetic manipulation, it is not a silver bullet. It has still got some limitations. It works very well only in those traits wherein loss of function is desirable. And we need to invest sufficiently, and we need to work together with policymakers in terms of promoting other conventional GM crops or transgenic crops. This was very, very evident in the recent statement by one of our advisors who is part of agricultural sector in Niti Aayog, Dr. Ramesh Chand. He has mentioned that oilseed security or security in terms of oilseed sector is only possible through GM crops. And we need to understand and appreciate this particular fact. And finally, I would also like to mention that biotechnology in terms of precision farming technology we need to integrate with other tools like data analytics, sensor systems, and then IoT in order to see that part of the solution can be deployed through biotechnological tools in terms of varieties with better nutrient use efficiency and others. But part of it should also come from other tools. And finally, one area of concern is that we have made significant gains or inroads in terms of policy decisions with regard to gene editing. But where we have failed is that we need to also address the IPR related limitations in this particular sector. And we are facing a lot of bottlenecks, not only our group, but those who have developed products through gene editing, they are also facing various limitations. And this is an area where we need to address them in the right perspective. With this, I stop my...
Thank you. Thank you, Dr. Sundaram.
Yes.
I think you covered a large ground: climate resistance, greenhouse gas emissions, precision breeding, genome-assisted breeding, the need to revisit functional genomics, the need to choose the appropriate technology. You can't use a spanner in place of a screwdriver. So there is no competition between transgenic crops and gene editing. They have to be appropriately used. We'll try to cover all this. There's a lot of fodder now for a detailed discussion. But I'm also aware of the clock. We have almost reached 30 minutes of our discussion. So almost 1 hour in our discussion, and we have another 30 minutes left. So I would like to make this remaining part a very interactive session. I would also encourage the viewers to post your questions on the chat box, Let me start with the first question that is posted on the chat box. This is by Bhavesh Palan. Bhavesh, thank you for posting it. I'll ask you a question on your behalf. What is your opinion regarding the licensing for the commercialization of gene-edited crops? I guess Dr. Bansal would be the appropriate person to do this. Now, just quick few thoughts from me. As a research project, you don't have to too much think about IP, but we are talking about application of this and commercialization. Now, do you have ways by which you can get around the two patents, the Broad Institute and Doudna's and Charpentier's two patents? We don't know which one. If you have to license it, from whom do you license? Is there any program by the government to actually get an overarching license and then supply it to people in India? Your thoughts, Dr. Bansal, on this specific question.
Thank you.
And
Certainly an important.
You can specifically address it to Mr. Bhavesh Palan, who is a participant in the meeting.
Right. No, thank you. And it's a good question, no doubt. And as you very rightly mentioned, Narayanan, that these two institutions actually speaking are holding this patent. But nevertheless, the Indian government is aware of it, that how important it is to have such a kind of a license, particularly as you need it for commercialization of these products. And government of India is making all efforts. I just can say only this at this moment. But therefore, important for us is to reach to that level of product commercialization stage. We must try to develop these products, get it through your IBSC, through your SOPs, which are there in the institution, and accordingly, we can then take it forward. So bring it to the level of field trials. And I'm sure, though there are difficulties, I must tell you, not only for India, even globally for some of our developing countries. And the license for agriculture is available with Corteva. And I think, again, some discussion was there before with ICAR and some more things are also happening. But at the same time, quickly, I would just like to add, and since I'm interacting with the group in Jennifer Doudna's group in IGI, actually they are developing entirely a new technology. I mean, this is only for this reason. There is a dispute somehow, unfortunately, going on, with regard to it. But then that institution is really aware of all these issues which we are facing currently in India and African countries, and they are working to come up with some kind of an alternative technology, and hopefully, we'll be able to really have it soon. That's what actually I can only say at the moment. Thank you.
So I can see Professor Sudhir Sapori raising his hands. Go ahead, Sudhir.
Yeah, actually, in response to this question, I just was listening to the seminar of Dr. Chinnaswamy, and he was talking about genome editing plans. He mentioned in the end that I couldn't get the more details, that government has this policy of getting one nation, one license for genome editing crop plants. And they already are working on getting, or they may have got, freedom of operation from UC Berkeley and MIT Harvard and other groups. So it seems this would be possible within the next few months. That's what he was mentioning. They are working on it, or they have already got it. Maybe more details Chinnaswamy will have. But
we are working on it, sir. Yeah.
They're working. Yeah, that's what he said, that one nation, one license will be done within a very short period of time.
Yeah, but the challenge will be, or the complication is, I'll come back to you, Sundaram. Which license, whether it is University of Berkeley, Doudna's license, or Broad Institute, the Chinese guy's license, because that itself is being disputed. So that's one complication that can happen. Anyway, it's reassuring that the government is also seized of this issue.
The government is seized of this.
And given the importance of this platform, we may have some options in the immediate future, I hope. Yes, Dr. Sundaram, any intervention?
Sir, I've been discussing both with the innovators and those who own the license, and also with those in India who have developed gene-edited products. There's a difference of opinion or a difference of contention between the approach of Government of India and what the developers of this particular technology, or rather those who have purchased the commercial freedom to operate license have it. For example, Corteva is ready to give access, like say they are ready to give us the freedom to operate for any product, but they want it on a case-to-case basis, because what I got to know is that an alliance called as Open CRISPR Alliance has been formed by several multinational companies along with the innovators, They don't want it to be licensed, want to give a freedom to operate license for any crop or any trait, because stewardship is also another question. They don't, for example, if somebody wants to reduce the nicotine levels in tobacco, that's a thing which they don't want to invest, because it can uh, sort of spoil the image of the technology.
Got your point. Got your point, Sundaram. I think,
So I think government of India should...
Bhavesh, as you have noted, there is no one answer to your question. In fact, people are still grappling with the situation, but hopefully this confusion will be cleared. But I am provoked to ask this question to the panelists. I have gone through the patent document, and most of the claims are based on the nuclease which is used, Cas9 or Cas12, the more recent ones. Because this system is so widely prevalent in the microbial system, can't India develop its own nuclease combination system? So, which can get around the patents that is already there. I think we should start at least thinking on those lines as well.
You're right, Narayanan, if I may.
A quick question, a quick answers from some of you, and then we'll move on to the next point.
Sir, one important issue what we need to realize is that there are two sets of patent applications. One are the foundational patents, which revolve around the whole technology or the process. Another is specifically with regard to the nucleases, what they have used. While we can just maneuver around the question of whether we use Cas9 or Cas12 or any other novel nuclease, that is something wherein we can develop some indigenous tools.
I find no investment is going into discover new nucleases. Anyway, I think let's close the discussion unless you have a very pressing point, Mr. Bansal.
No, you're right. I think it's important, and that's what we are giving this message to some of those youngsters and microbiologists. But maybe in the meanwhile, there is one other than SP-Cas9 available in India, in IGIB with Dr. Debo Chakrabarti, and that's also being tested in plants at the moment.
Wonderful, wonderful. So there seems to be some light at the end of the tunnel. It's all not darkness. So with a positive or an optimistic outlook, people should start working on these areas. So let's move on, because I see that new questions are being loaded in the chat box. But before that, I want to pick the brain of Professor Sudhir Sapori. You mentioned several things, amongst which you mentioned about creation of variability using genomic data and artificial intelligence. Now, crop improvement from the very advent of agriculture has been through the leveraging of variability that is either naturally existing or the artificially created, and then from that selection. So that is to put it in just two phrases. So these are new methods by which you can create variability. Now, there is a lot of talk about use of artificial intelligence and machine learning in agriculture as one of the technology platforms that will help in agriculture. And my observation is that many of these projects are championed by engineers and very, very smart people. But artificial intelligence or machine learning all depends on the data set that is created by the biologists in the learning system, so that artificial intelligence can really replace or at least partially supplement human intelligence and make analysis of large data possible. Now, is there sufficient emphasis on the biology part of creating these data sets, large enough data sets, so that the artificial intelligence can discriminate between the real effect and the noise that is around that effect? Are we spending enough time in doing that? Or are we actually being taken in by the glamour of some of these new technologies?
No, I think many of the groups are very huge groups where biologists and genome scientists and engineers and AI people are involved. This new platform, which I was mentioning, Evo2, this has been now trained on 126,000 genomes across the tree of life. It's one of the huge database on which this platform has been now trained. And they said with this kind of platforms, we should be able to do precise internal engineering things. So they were to because it's not any specific trait I'm looking at is not one gene mutation. I mean, because you have to look at everything. If you are going into the productivity, you have to look at flowering, you have to look at nutrient efficiencies, you have to use other areas, and then you get the kind of productivity. So these AIs can tell you which are the key markers on which you should be tinkering with yourself. Because right now our information is based on gene to gene basis,
Right.
and each one takes up his own gene, tinkers based on some information available. But I think we have to look at whole plant biology itself in order to make it a sustainable in future. So that's why these AI systems, when they are getting better and better, will be able to really give you information. In Arabidopsis, they have done. They are now moving from genotype to phenotype. They can predict these are the genes which you need to tinker with, and you will get this particular phenotype. These predictions have already been done. Now people are doing functional validations of this. I think this
Okay,
recent meeting in
nice to
Roundup,
know that. I
they
think
have,
so that it is reassuring. I just want to add that Bhavesh Palan, who asked the previous question, he has thanked all the panelists for the answers and also mentioned, which I wanted to reiterate, that we should make sure that the genome edited crops don't go the same way as the transgenic crops in this country. Thank you,
yeah, this
Bolin, for
is
cautioning us about it, and we are aware of that. Now, moving on, I think Dr. Vibha Ahuja, you mentioned about certain reforms needed in the IBSC committees for proper implementation of the regulatory reforms. What could be the potential pitfalls for somebody who is venturing into using some of these technologies and not getting caught in the web of regulatory morass? So, what is it that one should take in?
Actually, sir, more than the reforms in the constitution and all, I was referring to the capacity building of the people, because if
Okay.
you go to a university, we had Dr. Sundaram leading IIRR, himself a biotechnologist who understood the SOPs and everything very well. And he is the one who will take the decision that this is free from exogenous DNA and can be cleared. But if we go to the universities, to many other institutions, there are not that level of expertise in the regulatory committees. So we really need a kind of a big initiative to not only involve the PIs. If we really want to make, of course, licensing is a very different part, but in terms of the development, meeting the regulatory requirements and then reaching to a level where they can go to the farmers field. So this is one area where we thought we've ourselves seen that lots of, despite being having a clear cut policy, as Dr. Bansal mentioned, the regulations are there, this has been exempted, the guidelines are in place, the standard operating procedures are in place. But we have to also make sure that there's enough capacity to understand all those and meeting those compliances. We've had, unfortunately, a big lag in because of the negative, what I would say, the perception or the various decisions regarding the GM crops. So the whole system at the grassroots level, where actually things are going to happen now. If you see the new SOPs or the guidelines and the exemption, it is the institutional biosafety committees who have to take the decision that this is now free from this decision and ready to go to the field. So we need to really equip them. That's what I meant, the capacity building part
Okay.
of it, reaching
Thank
at
you. I think,
boundary level and so on. Thank you.
yeah, I think that, so creating awareness is what
Yes,
you find as a
awareness and
challenge.
also, about telling them about those
Okay,
nitty-gritties of
that
the
helps me
SOPs. Yeah.
segue into the next point I wanted to make.
Yes, sir.
I think creating awareness to start at the graduate level, not at the expert level.
Right,
And
sir.
in that respect, I want to recall what Professor Sapori said, that we have to have transdisciplinary approaches. And I also mentioned that agriculture today, and sustainable agriculture particularly, is going to be a confluence of multiple disciplines, not the old systems which are being taught in school. Are we teaching something about artificial intelligence at the graduate level or even at the master's level? Are we teaching something about machine learning? Do you know enough about drones and their use in agriculture? Do you know about genomics, data management? So are we geared? I think since we have very eminent educationists also in the panel, or people who have been involved with academic institutions. Uh, so I want to pose these questions. Can we start with Dr. Bansal? Is our curriculum ready to accept some of these new technologies, or equip people with the basic knowledge to leverage all these technologies, or at least work in teams which are multidisciplinary? They need not be experts in any AI, but they need to actually communicate their biology with an AI expert so that it works. I'll ask Casey Bansal and then move on to Sudhir Sapori and then to Sundaram and then to Vibhahuja.
Well, Narayanan, this is very important. Actually, these are 2 points which we're talking about it right now. One is that at the school level, are we talking about all these technologies, digitization and use of artificial intelligence, machine learning, etc. And second, you're talking about, of course, as Professor Sapori reminded us, the transdisciplinary, multidisciplinary approach at the professional level. I think these are both very important. I would just like to mention what I know is that National Academy of Agricultural Sciences organized, a kind of a brainstorming, a discussion on this, and the policy paper is out, I believe, and that is something which could be circulated and used by even for the school children to know more about it, that what's the future, these technologies hold. And at the ground level, which is certainly very important when we talk of communication and even prediction of any kind of a disease forecasting, even soil nutrients availability, etc. And there could be a team of or some of those graduates coming out after their BSc agriculture, after their school, college, education, they could be even trained, to do all that kind of use of drones, even for that matter, utilized, for predicting, what we don't know yet, and then we can have a solution ready available before any damage occurs. And of course, transdisciplinary approach is certainly very, very important. And, we're talking of not only here, plant biotechnologists, microbiologists are going to play a very big role as biologicals. It's biomanufacturing, as we're talking about it. Government of India has announced in August 2024 a Bio E3 policy. So I think the biologicals, the microbials are going to play a very big role. And that is something we should be moving ahead along with the plant genomics and all we're talking about it.
Thank you. I think so. In brief, there needs to be some changes in the curriculum or some additions. That's the import of what you said. Do you agree with that, Professor Sapori?
Actually, being involved also with the DAV schools and colleges organizations, I have been visiting many schools, different places. But very happy thing is that at school level, at 10th, 11th, 12th, that there are specific courses only on AI, ML, and other learning technologies. And I have seen some of the school children building up good platforms, based on AI. Only problem is when we go into higher college level or the universities, I don't think that is happening in terms of utilization for the biological systems at the moment. So I think there is some gap here. Initially, school level, now most of the good schools are teaching it. In fact, I was in one of the schools, which I didn't have one in JNU. They have 3D printing technologies. And that student demonstrated how he can use the 3D printing technologies. So I think there is a lot of things which are happening, but this needs to be carried forward, especially in the agriculture universities. You can think about when you bring it in the curriculum and into practicals the days. In fact, I was just to mention, Kailash probably knows, ICR has given almost like 500 crores for genome editing technologies, both for horticulture, crop plants, and fisheries. But I don't know the whole setup at the moment because this is within the ICR network, whether they are involving these people, agronomists, microbiologists, AI people, and so on and so forth. I think that kind of a network building within that whole thing should be done. I think it should not be left only to Sudhir Sapori kind of things who does a work in the laboratory itself. He has a gene and he will knock it out. But that kind of technology, it has to be much more broader, wider perspective to see if this technology, as she was saying, Narayan rightly pointed out, this may not land up into the same situation where we landed with genetic engineering technologies because we didn't really realize that what will happen downstream in terms of their performance in the field level or regulatory things and other things. So I think this needs
Thank
to
you.
be knitted very well together.
Thank you. So there are bright spots among the thing, but it needs to be spread into some DPS and allied schools may have it. Certainly in the graduate level itself, this needs to be introduced formally. And I think some of us, if Dr. Radana, Dr. Rajam, if you are going to give some recommendations to authorities from this, this can be one of them in terms of reforming the
Yeah, sure,
curriculum.
definitely. We'll pass on recommendation to the DBT.
Okay, I must, because
some
the
other
chat box
ICM.
is blazing with a lot of comments.
Yeah, I know.
So I must take some of them to be fair with them. Dr. Bansal and Dr. Sapori and others and Faba. There is a comment by Gayatri that a specific conference on the IP position for gene edited crops would be a welcome thing, because there are a lot of complex structures and people need to understand it better. Now here is a provocative question from Hemasri. She was wondering whether GM crops actually contribute to climate change. Sundaram, you want to take it first, and then we can go around the table.
I would definitely not agree with the contention, because, of course, there have been some concerns regarding reduction in biodiversity and other issues. But GM crops have made a positive impact in terms of mitigation of climate change. This is very much evident with regard to a single crop, that is BT cotton, wherein there has been a tremendous reduction in the application of insecticides. In fact, I would say that GM crops should be one part of the solution for mitigating climate change. And there are very good examples wherein a lot of innovators have developed very good GM crops, which are either drought tolerant or take up lesser. For example, one example I will say, of course, it happened way back in 2009, wherein one company called Arcadia Biosciences, in collaboration with UC Davis, they transferred a gene for nitrogen use efficiency, and they developed a wonderful rice variety which can grow under 1/4 or 1/5 of nitrogen dose. We saw it in field trials in California, but because of our policies, the product could not move ahead in India. In fact, understand that the product was licensed by Mahiko, but we couldn't go any further. And there is not enough proof to show that we lost our, where we are facing the problem of climate change because
Thanks,
of growth of
Sundaram. I think your
in
answer is an emphatic no. It doesn't.
Yes, sir.
Dr. Bansal, you have been a great advocate of GM crops. Responsible.
Right.
responsible
No, thank you. No, I think, I would.
Do you agree with Sundaram?
No, I will agree. I will make this statement that GM crops do contribute to climate resilience directly.
Resilience.
And I give you an example. Already a published study. I don't recall the name of the QTL, which has been taken from barley, which has been overexpressed in rice, with a substantial reduction in methane emission measured, over years. And I'll be very happy to share that paper. Directly, methane emission has been reduced. And in fact, that's because the exudates, coming out of those roots from the rice in the microbial anaerobic system, they're having some control over that. And therefore, and second example I'll give you, where Eduardo Blumwald, our good friend, actually in University of California, is working on it. He's actually asking the plants to develop, even in rice for that matter, produce those what do you call them, secondary metabolites, which are exuded by the roots into the soil, and they make a biofilm, and to induce, not only through nodulation I'm talking about, to induce otherwise free living nitrogen fixation in the rhizosphere, and that will cut down the, if it's indirect evidence, cut down the use of, nitrogen, and therefore it could be utilized further, for helping climate change.
Thank you, Dr. Bansal. In fact, I'll quickly add that
one thing which comes to my mind. Maybe Dr. Sundaram Madar can think about it. Developing genome editing plans or genetic engineering plans for organic farming. So under those conditions of organic farming, whether we can get the same level of yields what you will get under the chemical fertilizer situations. So I think that would be one thing where you can mitigate climate change issues.
But
it's a good idea. It's a good idea.
It's a good idea, but then
One could
genome
think
editing
about it.
should
I've
not
been
be.
thinking, but
there are people already thinking about genome editing should be excluded from organic certification.
Yeah, I think this would be something which people could try, you know.
We can have a
With-
different additional
With
planning.
the minimum inputs, you get the maximum output, and without
Yeah,
changing the
it's a
CO2
good idea. Good idea.
gases and all that. Yeah.
So the tyranny of the clock, we have hardly 10
Just
minutes.
one
left.
remark quickly, one remark on the comment made by Gayatri that we are already into organizing the kind of conference or workshops he's talking about it, and we'll be able to provide that information soon on this.
Okay, please note Gayatri. I think so. Let me call upon Soma Marla from ICR. Was she a colleague with you, Dr. Bansal, at NBPGR? Soma Marla is there. Can you unmute yourself? Okay, let's move on. Is she
Yes,
there?
I'm here. Good morning.
Okay. What is your question, or do you have any comments?
Yeah, my question is either it be GM crops or genome editing, we have been working on very narrow variation, a platform on a very lab model based systems only. And to widen the variation, we should really go for tapping the gene bank germplasm, especially mini cores, as suggested by our former director, Bansal. But the thing is, we have plenty of drop genomes already assembled, and we need to reassemble them and summarize them into pan genomes so as to widen the variation so that we could exploit variations from different environmental and soil and other aspects. So till then, the GM crops or the genome-edited systems will remain mostly lab-based and will not answer many environmental and health concerns. We need to go.
Yeah, I think your point taken. I think good point,
Okay.
Dr. Marla. I think Dr. Bansal also alluded to that. And not just based on phenotype, the variation should be characterized, but I would take the cue from Professor Sapori's intervention where he said that we should use genomic data and artificial intelligence-based methods to create or characterize the variation. So with that, we have 5 minutes left. I want to ask a quick question about
No,
Sundaram,
Dr.
you
Narayana,
mentioned-
no, Dr. Narayana, we have about 20 minutes because I've devoted 15 minutes for interacting with the speakers and the participants. So that is also part of panel discussion. So
Okay,
we can
I thought
continue
that-
till 11:15.
Oh, thank you for that. So
Yeah.
I
It is
thought
there
I
in the
had to
program.
close
It
it
is
at
there.
11.
It is there in the program also. Yeah.
Okay. So Dr. Sundaram, you mentioned about greenhouse gases emission control
Yes.
in rice cultivation. If I remember the statistics right, I think about 10 to 20% of the methane emission, depending on whose statistics you look at, emanates from the wetland rice fields. And one of the major problems of converting wetlands into drylands is the weed problem. And herbicide tolerance is a trait. We have a GM herbicide tolerance trait. We have a gene-edited version of herbicide tolerant trait. We also have a natural mutant, which is being promoted in the ICR system. How much do you think it would go to DSR, to give the all-encompassing term, dry-seeded rice, go to mitigate the problem of greenhouse gas emissions?
Sir, we need to understand that the three options what you offered, the GM rice or gene-edited rice or natural mutant available, these are all one of the several options to mitigate greenhouse gas emission. And already we are seeing tremendous positive impact in farmers' fields in the states of Punjab, Haryana, and then several other areas who are growing Robin mutant, conferring resistance to a herbicide. And farmers are able to clearly see the difference. But only one concern remains regarding deployment of herbicide-tolerant crops, whether developed through gene editing or through genetic engineering or through natural or artificial mutations is stewardship. That if that is taken care, I would say that it can go a long way in terms of converting a lot of area from wetland paddy to a direct seeded rice cultivation system. We need to remember that direct seeded cultivation is not a dry cultivation. There also we are going for an alternate wetting and drying. For some time we'll be impounding water, but it will be only up to the level of field saturation. And in that particular situation, the emission of greenhouse gases are very, very limited. And I think it's one of the positive interventions wherein through biotechnology, either through mutation breeding or through gene editing, we can certainly improve the tolerance of rice to various weedicides.
What do you think about drip irrigation in rice cultivation?
Sir, I'm cautiously optimistic because not all soil types are amenable for drip irrigation. We need light textured soils. Heavy soils will clog the drip outlets. So drip irrigation makes a lot of positive impact in terms of saving water and also partly mitigating the weed problem, but it's not suitable for all soil types in which rice is grown.
Okay, thank you. Now let me go to a question by Professor M.V. Rajam. I think this is to all panelists, so we can go around the table. Is there any hope for GM food crops being approved in India? And if so, if there's a promise, when will it become a reality? That's Professor Rajam. Yeah, you can go first, Dr. Bansal.
Yeah, there is certainly a big hope.
Only Dr. Bansal can answer this.
No, no, thank you. No, thank you very much, sir. Actually speaking, probably you all know we have been making struggle for the last more than 20 years. And just, hope sustains life. We believe in that as well. And I'm sure, while Government of India is putting all its efforts, and we are along with Government of India making our efforts, even the judicial system. And I'm very hopeful in this regard that yes, we will bring GM crops onto the farmers' field, particularly to start with after BT cotton, GM mustard. And Professor Deepak's Biotechnology, he himself is working so hard day and night, along with Dr. Vibha Ahuja and many others of his team, that certainly, I think this technology will see light of the day for sure, with about 25% more, production, at the productivity levels. And certainly we need more edible oil, production in our country because we're importing more than half of it to meet our requirement. So I'm very hopeful about it that more GM crops will certainly, find way into our farmers' field.
Sir, if I may add.
Yeah, please.
Yeah. So just to add to what Dr. Bansal said, and to this question, sir, that Government of India has given approval for GM mustard. It is only stuck in the court, Supreme Court, but that's also a very, very positive decision. As I said, the brinjal was not cleared by the government, but mustard was cleared by the government. And then, of course, we have these challenges in the Supreme Court. But I would like to draw your attention to
I thought brinjal was also cleared by the government. It was
by the GESE, sir, not by the Ministry of Environment. Ministry of Environment stopped it. No, the minister stopped. So the minister did not give clear. Here the ministry
mustard has cleared it. Okay.
Yes, sir. It's only in the court that there is the petitioners have filed to stop the trials, sir. So, sir, I would also like to take a minute here to draw your attention to what is happening globally. I mean, we really have no choice but to move also to use the GM technology, as was pointed out by Dr. Sapori, Dr. Sundaram, and all other on the panelists. And you see what is happening. All those countries who were little like naysayers, were in that similar group that, oh, whether we should not. And the most prominent of that is China. It's very, very important to know that in the last 1.5 years, they have gone ahead and they have approved almost, I think till the end of last year, it was 81 different varieties of GM maize, GM soybean, cotton, of course, was there earlier. And they are going on a big way, not only the products which were brought in for which the applications by the multinational companies, but their own institutions. So we are seeing a lot of change, sir, at the global level. And the Africa. In Africa, all the new type of crops, whether it's Nigeria, Kenya, yesterday it was Ethiopia, all these people are going on for newer crops, the GM food crops actually. So both the maize, there was a lot of resistance earlier, even in Asia, sir, Indonesia, sugarcane, so many of them, So the situation is changing, and I think the government in India is also very well aware of this fact, and we see a lot of positive, approach to the whole thing, and I'm very sure that the GM food crops will be in farmers' fields very shortly. We have got, sir, so many trials started now of the food crops from the public sector institution, including the ICR institutions.
Thanks, Dr. Ahuja. So the global situation should be a positive incentive for us also to move quickly in this direction.
Thank
You
you for
have
the
any? Yeah. So I hope, Rajam, you are satisfied with the answer.
Yeah, very much. Thank you.
Now, my last question is on field biology. I think this was also raised by Professor Sapori. So far, variations has been based on tapping lab models. How important is it to integrate it with field biology? You may want to explain first, Professor Sudhir Sapori, and then we will go around the table.
I think problem, last year, last year, I think the EMBO.
The reason I wanted to talk about is many people or who used to pursue biotechnology look at rice plant as in a microfuge tube. Whereas out there in the field, it's very different.
Absolutely,
But I think the perception is changing, but I would certainly like to hear your views on that.
I was mentioning, that last year in the EMBO meeting, the chairman asked the scientists, if you want to do good science, move out of the lab. And I think the situation in the field conditions is absolutely different from what we create within our laboratory systems or even in the greenhouse condition. For example, when I do a genetic engineering plant, even my own Glaxless, I'm talking about it, we test it in a pod system where we have provided the best conditions of soil conditions, we water, we give a very nice environment of air conditioning and other and so forth. And we say that yes, this gene function is something which gives you a specific trait. But the whole situation changes when we do testing outside. So therefore, it is very important when we do even transcriptomic studies under uh, laboratory conditions, these have to be done under the field conditions. And this was done for sorghum, many papers now coming up in PNAS and others, where they did all transcriptomics gene regulation studies in field conditions under the normal environment where they are normal, these plants are growing. And of course, these are huge investment projects because you have to do all at different stages of development. And from there, the data which they obtained, they found that it was the expression of profile was totally different from what they were getting in the field lab conditions. And that data is very, very important, which then should be used by AI and others. I think that's where the quality of data based on which AI will make a decision becomes very important. So I personally feel the field studies should be carried out for genomic level, getting the data from the genomics, and that would be a fodder on which our AI or ourselves can decide which genes to tinker with through genome editing or any other technologies.
Thank you.
So
Thank you
I
for underlining the importance of field studies.
ecological genomics, that's what they called as ecological
Okay.
genomic studies, which has to be taken up in future, I think.
Okay, that's a new term I'm hearing. Thank you for underlining the importance of field studies. Dr. Sundaram and then Dr. Bansal. Quickly, do you agree with Sapori or you want any other perspective to be added?
I totally agree with Dr. Sapori. I would narrate our own experience with regard to selection of the best event among our gene-edited lines. When my colleague Dr. Satyendra had generated about 80 stable events through editing a single gene in the background of Samba Masuri, what we did is we planted all these 80 events in stripped rows in our transgenic greenhouse. Fortunately, we have a DBT approved biosafety facility wherein we can grow the plants in near natural conditions in a greenhouse or a net house. And what we were able to see is that we involved the breeders and also our agronomists to select the best possible event. And fortunately, we were able to identify one best event which had magnified phenotype with regard to what we were expecting. And when that particular event was finally approved by RCGM of DBT for open environment, or say open environmental release, it happened to be the best event. But there was also a lot of opportunity for us to learn from the other events. And I think unless whatever technology we will be developing in terms of developing an improved project, they need to be brought to the field and they need to be tested along with the other entries. And I totally agree with Dr. Sopori's perspective here, sir. Thank you. Sir, you are muted, sir.
Am I unmuted now?
Yeah.
Dr. Bansal, any other thoughts, or you agree with
I couldn't agree more with Professor Sopori, and that's what we have been also advocating and talking about it as physiologists, or crop physiologists, that we must go out into the field to test it. And in fact, apart from the study and kind of conference EMBO, Professor Sopori is talking about it, there was a paper by, I think, some 20 best, scientists, bunch of scientists, 20 of them, I think about 5 years ago, when we were doing a single gene and phenotype, and we say it imparts tolerance to drought, salinity, etc., they cautioned actually all the researchers, that stop making all these claims and better go out into the field and then test it. So therefore, I think this is very important that we must continue, teaching our own younger colleagues, that unless you have a proven effect right under targeted field environmental conditions, we cannot claim that we have a product. So it's important to go out of the lab, as Professor Puri reminds, and do better science. Thank you. Yeah.
So I think all of you have emphasized the importance of field studies. I completely endorse that, and I agree with all those points. So we have had a wonderful discussion. I think the last 5 minutes, I will leave it to the organizers, Professor Rajam. Before that, I leave that. I want to thank all the panelists for the wonderful discussion, and also all the participants who have posted a lot of questions in the chat box. I couldn't, I'm sorry, I apologize for not taking all the questions.
Yeah.
Dr.
One
Narayana?
point I would like to make, Narayanan, if you allow me.
Of course, you can make the point. There's 5 minutes for you.
Yeah, yeah.
I know,
So,
I think the next speaker has not joined. We can still continue.
yeah, yeah. So, the point is what Dr. Subodhi mentioned, that multidisciplinary approach is required to address the problems in agriculture, not only agriculture, in any other discipline also. And also the point that you all made, that that kind of curriculum is required in colleges and universities. So in that connection, University of Hyderabad started what do you call it, as integrated master's program of 5 years after +2. They undergo the training in mathematics, statistics, computer sciences, physics, chemistry, economics, everything in the first 2 years, and then go to different disciplines. Either it is biology, it is chemistry, or physics. They go in all areas, and the graduates are coming. The point here is where are the opportunities for this one? Agricultural Council says only agricultural graduates have to be employed in ICR institutions. Veterinary Council says only veterinary graduates have to be employed. Medical Council says only medical graduates have to be employed. And where is the opportunity for these? And as a result, none of these experts, either in Agriculture Council or in Veterinary Council or in the Medical Council, they cannot address the problems in today's biology. They need the expertise in different disciplines, as pointed out by the experts. So this is the major problem in the education system in the country, which needs to be addressed if we have to make any kind of developments or improvements in the science and technology. This is the first thing that we need to address.
Well, Dr. Radhana, you made a very significant point as a long-time educator.
Yeah.
It's very important. But the problem is that everybody wants to protect their own turf. And so this may require a wider discussion with an open mind.
Yes.
We have to, unless anybody has any quick solutions for it.
Sir, as far as ICR is concerned, it's little bit more liberal. Dr. Bansal will agree with me. Nowadays, in various disciplines, people who may not have done their graduation in agriculture or post-graduation in agriculture, they are also encouraged to apply. And I would say that there are at least 15 out of 40 or 45 disciplines in agricultural sciences wherein people who may not belong to agricultural stream, they are encouraged, sir.
Good,
Of
good.
course, we need to do a lot more in this regard, I agree.
The new education policy actually emphasizes this, that you must get courses other than your own,
Yeah,
family.
education policy is okay, but
But it is
the
not
policies
everywhere.
of these institutions have to be changed.
Well,
I
I
know
can also add that having industry faculty as adjunct or part-time also needs to be encouraged or more widely adopted so that you get a commercialization perspective also in the whole education. A lot more need to be done, but I don't think we can solve all those issues in this panel discussion. We will take it up, but some of the people who can influence the opinion are here in this panel. So they will take the message out and go. It's almost
one of the
1 minute.
recommendations here, yeah.
So it's only 1 minute to
Professor
11:15.
Attapalli Reddy is raising.
Yeah.
finger.
What
No, I have to.
Red is right. We had one very bad experience in our university where our plant science students were not recognized for any botany lecturer's post in Public Service Commission. They said plant science is different from botany. So
Well, that's fine here. I think let's not discuss specific examples.
yeah.
Then that will go on.
we-
I think generally the point has been made. I think we cannot sit in silos. It needs to be opened
Yeah.
up. That's the key, I think, in all dimensions. And Professor Sapori mentioned that the new education policy has got some steps in that direction. But we all need to work together. And if specific recommendations on this has to be made, we can actually come together again with specific theme and agenda for making those representations. You know, Dr. Rajam, why I was in a hurry to close this meeting at 11:15. I had to go at 11:30 for another meeting.
Yeah.
So
the next speaker
I am
has not
done.
yet
So
joined.
I just want to thank all the panelists and the participants who very actively participated. Again, once again, apologies for not considering all the inputs in the chat box. Some of it could be answered on a one-to-one basis. And Professor Rajam, as the organizer, I would suggest that you can collect all those questions from the participants during the plenary sessions. And some of us can respond to some of those questions if it is directed specifically. I can see Ram also in the picture. That means he has just joined. So you have the next speaker for the next session. Thank you very much.
Good morning, everyone.
Thank you. Thank you.
Thank
You
you.
know,
Thank
that
you very
like to
much. I
thank,
think for all
Thank
the
you,
Thank
thank
you.
you, Dr.
I
Narayanan.
would like to
for
thank all the
doing a great
all
job,
the
and Dr. Rajam, thank you all. I take
I would
leave.
like to thank
Thank you.
all the panelists, for taking your valuable time.